In the early 1950s, the scientific community was engrossed in the enigmatic structure of DNA, deoxyribonucleic acid. In the decades leading up to Watson and Crick's work, many prominent scientists have made important advancements regarding the nature and function of DNA. In 1869, Friedrich Miescher discovered a substance in the nucleus, which he provisionally named nuclein. We now know this substance as DNA. In 1919, Phoebus Levine further identified the components of the molecule: nitrogenous bases, ribose sugars, and phosphate. In 1927, Nikolai Koltsov suggested that two minor strands comprised DNA, and soon later William Astbury produced the first X-ray diffraction patterns of DNA. Some important vocab includes crystallized DNA. These are DNA crystals that form when a double helix is suspended in liquid that evaporates. They grow in patterns dictated by the information stored within the strands. X-ray diffraction. This is the scattering of X-rays by the regularly spaced atoms of a crystal, useful in obtaining information about the structure of the crystal. A helix is an object having a three-dimensional shape like that of a wire wound uniformly in a single layer around a cylinder or cone, as in a corkscrew or spiral staircase. Watson and Crick made an important distinction between right-handed and left-handed helices. If you hold it pointing away from you and it twists clockwise moving away, it is right-handed. Otherwise, it is left-handed. Francis Crick was born on June 8, 1916, near Northampton. He studied physics at the University College London, and during World War II worked for the Admiralty on the development of mines. He changed from physics to biology, and in 1947 began to work at Cambridge University. James Watson was born on April 6, 1928 in Chicago and studied at the University of Chicago, Indiana, and Copenhagen. He then moved to Cambridge University. Watson and Crick worked together on studying the structure of DNA, the molecule that contains the hereditary information for cells. Watson and Crick hypothesized that the structure of DNA had two helical chains each coiled around the same axis, as shown here. They used balls and sticks in order to create real 3D models of their theories and findings. In order to support their hypothesis, they used many contemporary researchers' experimental results and findings. Watson and Crick probably owe most of their success to the results of two separate experiments, one carried out by Erwin Chargaff and the other by Rosalind Franklin and Maurice Wilkins. Using paper chromatography and an ultraviolet spectrometer, Chargaff found the base pairing pattern that DNA follows. He determined that although different organisms' cells contain varying amounts of DNA and different nitrogenous base ratios, the amount of adenine found in DNA would always equal that of thymine, while guanine and cytosine would always be found in equal amounts as well. Watson and Crick used this information to deduce the function and location of the nitrogenous bases in DNA. Likewise, Rosalind Franklin's experiment, Photo 51 in particular, further supported the hypothesis of DNA's double helical structure. This image, obtained through X-ray diffraction, helped determine the structure of a crystal by shining X-rays through them and recording their path on photographic film. The complex patterns that form on the film thus revealed much about the structure of the crystal studied. To the naked eye, the photo looked merely like an X diced up into bits, but to Franklin it confirmed what she and other scientists suspected: DNA was a double helix. DNA, as several other biological molecules, can crystallize, and using this method, Franklin and Wilkins obtained Photo 51. From this data, Watson and Crick arrived at a series of conclusions regarding the structure of DNA. They said, quote, "We have made the usual chemical assumptions, namely that each chain consists of phosphate diester groups joining, D-deoxyribofuranose residues with 3 to 5 linkages. The two chains, but not their bases, are related by a dyad perpendicular to the fiber axis. Both chains follow right-handed helices, but owing to the dyad, the sequences of the atoms in the two chains run in opposite directions. The novel feature of the structure is the manner in which the two chains are held together by the purine and pyrimidine bases. The planes of the bases are perpendicular to the fiber axis. They are joined together in pairs, a single base from one chain being hydrogen bonded to a single base from the other chain, so that the two lie side by side with identical z-coordinates. One of the pair must be a purine in structure, the other pyrimidine for bonding to occur." Watson and Crick, along with Rosalind Franklin, Maurice Wilkins, and Erwin Chargaff, have contributed to the discovery of DNA structure and have helped to inform years of research and knowledge necessary for progress.